So over the next couple of videos, as we're talking about the different stages of cellular respiration, you're going to want to start asking yourself for each one of these stages, what goes in and what comes out. Because I don't expect you to memorize every step. Each one of these stages has 10, 15 steps to them, and you don't need to know every single chemical reaction. But you do need to know what goes into each stage and what goes out of each stage. So you should create a table for what goes in and out for each stage of cellular respiration. So one row for glycolysis, one for the breakdown of pyruvate, one for the citric acid cycle, and one for oxidative phosphorylation. So you're going to have a table like this. And again, you can also think of this as input and output. What molecules are going into it? What molecules are coming out of it? And when you're answering those questions, make sure you think about two things. What's happening to the carbon and what's happening to the energy? And I'm going to always go over this in the videos, but you want to organize it for yourself in a nice table because this is some of the key information that you need to know about cellular respiration. So we'll start with glycolysis. So glycolysis is going to break down glucose into two pyruvates. Glycolysis can actually occur with or without oxygen, and we'll see that some organisms have the ability to do glycolysis even when no oxygen is present. Glycolysis is also identical in virtually every organism on Earth. So this is interesting because this really tells us that glycolysis was one of the earliest traits of primitive cells and that we've all continued to have that trait because it's essential, even though we've diversified greatly from those first original primitive cells. Glycolysis occurs in the cytosol. So even though the majority of cellular respiration eukaryotes will occur in the mitochondria, it's all going to start in the cytosol. There's three basic phases to glycolysis. So there's 10 chemical reactions, but we can break them into these more simple categories of steps. And so remember, you don't need to remember every single one of these 10 chemical reactions, but you do need to know the big picture of what's happening in glycolysis and what goes in and out. So we'll start with the energy investment phase. So glucose is going to start out here and it's going to be modified using ATP. So this is called energy investment because it's actually going to have to use 2 ATP. So you've probably heard you can't make money, or you have to spend money to make money, or you have to invest money to make money. The same thing's kind of true in the cell. It's going to have to invest ATP so that later on it can make ATP. So it's going to invest these 2 ATP. When it spends ATP, it makes ADP. So remember, ATP is always cycling between having the third phosphate and not having it. So when it spends ATP, it makes ADP. So in the second sort of phase of glycolysis, cleavage occurs, and to cleave means to split. So this is where a 3-carbon molecule, which drawing out here is little dots, splits into 2 of these 3-carbon molecules. And then the last step is energy liberation. So this is where energy actually gets released. And so what's going to happen here is it's going to make 4 ATP. In order to make 4 ATP, I have to use 4 ADP. Also going to make 2 NADH. In order to make NADH, I have to use 2 NAD pluses. Okay, so we take a look at the figure here. Here's the here's the glycolysis, here's the energy investment. So I can tell that ATP is being used by the direction of the arrow. So ATP is coming in and being used. Then I'm getting cleavage, so separation of a larger molecule into the 2 3-carbon molecules. Now each one of these has to go through this, so we can imagine that each step is now happening twice. This is going to be true for a breakdown of pyruvate in the citric acid cycle too. So when each one of these goes through these reactions, it's making NADH and making ATP and making another ATP. Same thing here, making NADH, making ATP, making ATP. And I know that it's being made because of the way the arrow is pointing out. So in these diagrams, the arrow pointing out means that molecule is being made or released. So in summary, I just want to make sure we're clear about inputs and outputs. So input and output, and again, remember, you want to ask yourself, what's happening to the carbon? So if I start with the carbon, I have glucose coming in. At the very end, I have pyruvates coming out. And there's 2 of them because there are 3 carbons each, and glucose is 6 carbons. Then I want to ask myself, what's happening to the energy? So input, I had to put in 2 ATPs. And to match that up with an output, that's going to put out ADPs, right? I'm going to make 4 ATPs, but I'm going to have to use 4 ADPs to do that. And then I'm going to come over to the right here because I'm running out of room. I'm going to make 2 NADHs, so I'm going to have to use 2 NAD pluses. So these are all energy molecules, so that's why I said what's happening to the energy. So some of the energy that was in glucose is now in these molecules. Pyruvate still has more energy in it, so it will have to go on to the next stages of cellular respiration. NADH has energy in it that the cell wants to convert into ATP, and so it will have to go on to oxidative phosphorylation. So cellular respiration is really a giant redox reaction. So if I just write out, I'm not going to balance the equation here, but if just write out the basic steps or the basic process. Okay. Oh, okay, I'm just going to ignore that here because it's going to turn off this drawing here. So let me just finish my thought here since you can still kind of see it. This is a giant redox reaction. The glucose is being oxidized when it's turned into CO2. And so when we're looking for redox reactions in cellular respiration, we're just going to look for NAD plus and NADH. So in glycolysis, I know that NAD plus was reduced to NADH. So that's just something you need to memorize. Remember, we talked about that in the previous chapter. And so if I know that NAD plus was reduced to NADH, then glucose in glycolysis must have been oxidized. And so I'll stop here.